Hello everybody and welcome to this If Oxford event, Chemistry Confections, the savoury edition. My name's Dane, I'm the festival director, and it's a treat to be introducing you to Jess Crompton, who is a researcher at the University of Oxford in the chemistry department. So she spends all of her days fiddling around with chemicals, but then we all spend all of our days fiddling around with chemicals, whether we're making food, whether we're looking at things in the environment, or even just breathing. So we just want to highlight some interesting aspects of chemistry today and start some conversations with you about your cooking habits and techniques, maybe doing some myth busting or offering some suggestions on what to do. But it's a pleasure to pass on to Jess, who I'm going to ask you to introduce yourself, explain what you do, and then we can join the journey of Chemistry Confections Savoury. Over to you, Jess.
Okay, thanks, Dane. Hi, everybody. As Dane said, I'm Jess Crompton. I am an experimental organic chemist, and I will come back to what organic chemistry is a bit later on. I've just started the second year of my PhD and the first year of my final PhD project. And I also studied my undergraduate degree, chemistry at Oxford University, as well as my PhD. So I'm going to share my screen and hopefully... Today we're talking about savoury food and the sort of chemical compositions of that, and hopefully shed a bit of light on the chemistry that's going on in cooking. So to start us off, chemistry really is everywhere. Everything we do in everyday lives is a result of some form of science. And specifically, chemistry has a massive impact on everyday things we might not necessarily connect to science. So here's some examples. Hopefully the top example is kind of your classic lab-based chemistry, which we would hopefully connect to science. But there's also important aspects of chemistry in the oil and petrol industry, in sort of the cleaning industry or the fast-moving consumer goods industry, in the pharmaceuticals industry. These are all not a giant leap from being connected to chemistry, but there's also some important chemical aspects in sort of the skincare and beauty industry, in the paint and pigments industry, and also in the power industry in the form of batteries. There's a nice link to Oxford here in that a couple of years ago, John Goodenough won the Nobel Prize in Chemistry for the development of the lithium-ion battery, which I think was really special because that's the battery that powers our mobile phones and our laptops. So it's a battery that everybody carries around with them on a daily basis. But what we're going to focus on today is the importance and aspects of chemistry within our food. So first of all, we're going to go back to school for a little while. I hope it won't be too boring for you. So the sort of classic image of chemistry that most people will have will be of the periodic table. This is the most iconic sort of aspect of chemistry. So I mentioned earlier that I'm an organic chemist. So what does that mean? That means I'm interested in this atom here, this element, carbon. I've got the whole table to play with, and I play with mostly carbon. I'm really interested in making carbon-carbon bonds. So carbon is for organic chemistry. Everything else on the periodic table and the chemistry associated with these elements is inorganic chemistry. Some people would say that makes organic chemistry a bit boring, but there's a massive amount of diversity you can get just with carbon and the elements bonded to it. So for food-related chemistry, the vast majority of that chemistry is organic chemistry, and the elements we're going to focus on are popping up now. So we've got hydrogen, which is carbon's best friend, nitrogen, oxygen, and a little bit of phosphorus and sulfur as well. The other elements are much less common in foodstuffs. And as we'll see, there's a massive amount of chemical diversity that we can obtain from this relatively small subset of the elements. So I'm talking about chemical diversity. How do we get there? Now we get there because the atoms can bond to each other. So an atom is the smallest subunit of an element you can get and still call it that element. So I'm hopefully going to turn my screen back on, if that's all right, Dane, because I could do with showing some models.
Go ahead. Yeah,
I'll keep it on for this little bit. So here we have at the bottom of the screen, when I've found my laser pointer again, and here we have acetic acid. This is vinegar. This is what you put on your fish and chips. And here is a sort of much bigger version of the molecule, but a model to help you understand what I'm talking about here. So as you can see, the atoms are bonded to each other, and we represent this with a line between the two elements. So here you can see we've got a hydrogen bonded to a carbon. Hydrogen is the white atom and carbon is the black atom. They're just sort of standard colours. They're not really white and black, but this is what we colour code them sort of in our heads for our sanity as chemists. Then you can see we've got a carbon bonded to a carbon. And then we have a double bond, so 2 bonds to an oxygen, which you can see there. I've got 2 bendy bonds here to an oxygen. And then another bond to a single oxygen and to a hydrogen on the end. This is an acid, which I will come back to. But because we are really lazy as organic chemists, and we often are drawing much more complicated molecules than acetic acid, we have our own shorthand. And I'm going to explain this to you, because if I tried to draw out some of the molecules we're going to talk about later, it would take me all day to draw them in this form on the left-hand side. So what we do is we say that if there is only a carbon here, it's got to have 3 hydrogens. So we don't draw the hydrogens if they're attached to a carbon. We just assume that they're there. So we can see here 3 hydrogens, but we only draw the carbon. We draw the carbon-carbon bond with a line. We don't draw the big C's. That takes time. And then we do draw our elements for our heteroatoms. That's atoms that aren't carbon and hydrogen. So we do draw the double bond to an oxygen and the single bond to an oxygen. And we do draw hydrogens that aren't attached to carbon. Hopefully that makes sense to everybody. Also, that molecule is quite flat. Most molecules are not flat. So here we have, let me get it in the right orientation for the matching of the screen. Hopefully you can see there. This is menthol. And you can see that I've got some of these bonds are different thicknesses. So the fat bonds here mean that the group of atoms, or the atom, is coming towards you out of the page. So you can sort of see here that this CH3 group at the top that I'm twisting around is coming towards the screen, because you can see that there's a hydrogen on the back coming towards me. And similarly, you can see you've got a hydrogen on the front here, and then this sort of branch group at the bottom is coming back towards me. There's sort of the plane, and you can see that that's coming towards me, and this is not. Similarly, this is a bold bond. So this bond to the OH group is coming out towards you. So that's how we sort of represent 3D structures on 2D pages as organic chemists. We also use this sort of strange chair notation because the way that this ring sits is a bit puckered, and I've got that upside down. So that should be how you see it, I think. Not quite. Yeah, so it's a little bit puckered, as you can see in both the way I'm holding this and the sort of computer generated diagram I've got here. Hopefully that all makes sense to everybody. And I'm going to move on to what is actually going on in the molecules. So to be an organic chemist, as I've just mentioned and shown with our models, you've got to be able to sort of picture the molecules in your head and spin them around, so you've got a good understanding of what they look like and how they behave. So what determines the molecular properties and reactions and how these molecules behave is the way in which the atoms are bonded together. And that's called a functional group, and it determines both the physical and chemical properties of molecules. We'll come back to what physical and chemical properties are. So these are different functional groups. R is just another carbon-containing group. We use it mainly so that we can focus in on the area of interest within a molecule by saying sort of this bit doesn't matter, this is the important bit. So we have alkanes. These are things like are present in crude oil, sort of long chains of carbons bonded together. We will see some of these later in our food molecules. Similarly for alkenes. Alcohol, this isn't just the alcohol that you might see in the shops or your parents might drink. This is any compound with an OH group on it. So even menthol that I showed before, this is an alcohol, even though this is not the same as what would be in a glass of wine. An amine is kind of the nitrogen version of an alcohol, but has its own set of properties. These halides, alkyl halides, they're called, use the group 7 of the periodic table, fluorine, chlorine, bromine, and iodine. They're very reactive. We don't see many of these in our foodstuffs. We're not going to find them in the body. They're just too reactive. And then we move on to these sort of double bonds of carbon to oxygen. There's a lot of different compounds in this family. We have aldehydes, ketones, acids, which we will talk about a bit more later. Esters have quite often have a very nice smell. There's one ester, I can't remember which one it is, but there's one that smells like pear drops, the sweets, and that's quite nice. And amides are a very key bond and functional group that is very key in our bodies and in our foodstuffs, and we will talk about these later. And arine functional groups are basically just big balls of grease as far as we're concerned today. So as I said, functional groups are responsible for the physical and the chemical characteristics. So the physical characteristics will be if it's a liquid or a solid as a bulk material, or if the molecules sort of like to be around water molecules or do not like to be around water molecules. So if they like to be around water molecules, they're described as hydrophilic. And these are some examples of these molecules that are hydrophilic. And if they really don't like to be around water, it's usually that they're very greasy, and they will be described as hydrophobic. And we will use these terms quite a lot in the talk today. The chemical properties, functional groups basically are formed when other functional groups react together to form them. That sounds a little bit convoluted, but basically the functional groups that you've got determines which reactions can take place within your reactants or your compounds. So to get us started, to understand about the chemistry of cooking, we need to know a bit about the compounds within our foods. So hopefully everyone's heard of the 3 main food groups: carbohydrates, proteins, and fats. But what's actually going on a molecular level with these food groups? So we're going to start with carbohydrates. Now, carbohydrates are a group of chemicals called polymers, and that means that they're made up of smaller subunits called monomers. And for carbohydrates, the monomers are simple sugars. So you can see here that I've got lots of these hexagons all joined together. They might all be joined in a long line like this. They might be branched off, like in the paler green. But this is the polymer, and the monomer will be sort of these types of simple sugars. There's lots of different versions of these sugars. This one is glucose, which is about the most basic sugar we can get, to sort of explain this. So when they're joined in long chains, they're found in cereals, and then sort of on their own, either one sugar or a couple of these joined together would be sort of more sugars that you would find in like your table sugars or sweeteners. So proteins are also really, really important for understanding what goes on in our food chemistry. And proteins are also polymers, but the monomers, so the smaller subunits this time, are called amino acids. And I've drawn you an amino acid here, a generic one. So as the name kind of suggests, we've got an amine group at one end. This was one of the functional groups I showed you earlier. And we've got an acid group at the other end, which was another functional group I showed you. In this case, there are about 20 different naturally occurring side chains. We will talk about this a lot more in the sweet session next week, if you're joining us for that. But for now, R is just a side group that we don't really care about too much. So when all of these amino acids are joined together, we get a long peptide chain like this. And then this chain will sort of fold in on itself to give this specific 3D structure or tertiary structure. And with proteins, the function of the protein and the structure are really closely linked. So if it loses its structure, it loses its function. And this is a reason for a lot of processes within cooking. Moving on to the last main food group, we've got fats. So we have simple fats. Now, in contrast to the proteins and the carbohydrates, which were polymers, simple fats are molecules. They're big molecules, but they are discrete molecules. And they're made up of 2 components, a fatty acid component, which are these sort of long wiggly tails here, and a glycerol component, which I've represented as this purple blob. So the purple blob actually looks like this in terms of skeletal formulae, which was our shorthand notation that I talked about. And our fatty acids look like this. Now you can probably see why we do this shorthand, because if I had to write C and then 2 H's on every single kink in this chain, I'd be here forever. So there's different types of fatty acids. There's unsaturated fatty acids. There's saturated fatty acids, there's monounsaturated fatty acids, and there's polyunsaturated fatty acids. This is linked to whether you have unsaturated fat or saturated fat, is determined by which fatty acids you've mainly got in your fat. So this is triglycerides, these fats compounds, but we also have, and this is what it would look like, sort of putting my schematic at the top onto the actual structure. We've also got phospholipid molecules, which are very important. These molecules are a bit like our triglycerides, but they have an additional component, which is this group here, which I've represented as a blue blob. This is a phosphate group. This is a very hydrophilic group. This really likes to be around water. In contrast, the big fatty acid tails, as hinted by the name fatty acids, are very greasy, and they like to be in sort of oily or organic media, so they are hydrophobic. So this molecule has a hydrophilic head and a hydrophobic end, and therefore we call them amphiphilic. And because of this, they have special properties, and sometimes they'll line up in special ways. So like this, we've got, this is a bilayer. So we have the top, we have all the polar head groups, so all the phosphates lining up on the top and the bottom, and the fatty acid tails sort of seeking refuge in the middle. They'd form this kind of arrangement if you're in sort of a water environment. So this is what it actually looks like in cells. Your cell membranes are made up of these. Also, we'll talk about a little bit later, and they can form these sort of vesicles or micelles, where you'll have like an oily substance in the middle, and then round the outside you'll have the watery substance. So you've got the hydrophilic head groups sticking out into the water, and the hydrophobic tails sticking into the oily or greasy layer. Okay, so today we're actually going to look at the science of chemistry behind cooking via focusing on one key make, which today is going to be a veggie supreme pizza. Now we're going to start a bit weirdly at the end, and we're going to talk about dips and dressings. So if you're a bit fancy and you're going to have your pizza with a salad, you might put a vinaigrette on your salad. If you're slightly less fancy or just really like garlic mayonnaise, you might want to have garlic mayonnaise with your pizza. So we're going to talk about the science behind these dips and dressings. So I'm going to start with the vinaigrette, and we're going to talk about oil and water. So hopefully you are all aware that oil and water do not mix. To prove this to you, I have a bottle with oil in the top and water in the bottom, and I'm going to shake it, and then we're going to leave it, and you will see that they will not mix. They will separate out. You can't really see, but hopefully you could try this at home. This is a very simple experiment. You can see you get lots of little sort of globular droplets that will then separate out into two layers. We'll come back to my bottle of water in a little minute. Now, the reason that they don't mix is there are no favorable interactions between the greasy oil molecules and the very polar water molecules. So water is hydrophilic, basically by definition, because hydrophilic means it likes water, and like likes like in chemistry, and oil is hydrophobic. So these unfavorable interactions basically mean that they don't want to be together, and they will minimize the area of contact, which is why I said we got lots of spherical globules when we first mixed it up, because if we have a sphere, we minimize the amount of surface area that has those unfavorable interactions. And eventually the globules will all join together, so they're bigger, so you have even less surface area exposed to the unfavorable interactions compared to the volume inside the globule, and eventually you just get two separate layers. And hopefully now you can see we've got our oil back at the top and our water at the bottom, so it's already separated in the time it's taken me to explain that. Okay, so with a vinaigrette, what determines where a compound dissolves? So we've got our oil layer at the top, and we can see that anything that is sort of oily or very organic, which is most organic compounds, will go into our oil layer. So anything that's sort of alkane, greasy, as I said, most organic compounds, any other sort of fats will go in there, because oils are basically, probably look fairly like this. I don't know exactly like this. This is just one I've made up really. But what goes into the water layer? So the water layer is anything that has a charge, and some other organic functional groups. So alcohol's quite like water, amines quite like water. They've also got a habit of, they will pick up, if there's any sort of acid hanging around, they will pick up H plus and become charged, and then they really like water. And the reason why charged compounds like water so much is water has a bit of a charge, so that the oxygen atom has slight negative character and the hydrogen atom has slight positive character. So in our vinaigrette, you'll have your vinegar, or your acidic compound will reside in the water layer, or form part of the water layer, because it's partly charged and is therefore quite polar and stabilized by the water layer. But we noticed when I did this that we have the oil on the top and the water on the bottom. So what determines the layer order? And basically it all comes down to something called density. Density is basically how much stuff you fit in a certain space. So as you can see here by this, these cubes, the cube on the right hand side has lots of green balls in a certain space, whereas the one on the left has fewer green balls in the same space. So the one on the left is less dense and will be lighter as a box to lift if they all weigh the same, which they do. Hopefully you've seen this sort of phenomenon in sort of what floats and what sinks. So cork has lots of big air bubbles, so it's a lot less dense than water, so it floats on the top. Wood, again, has a few air bubbles, but slightly more dense than cork, so floats. And then aluminium, there's no air bubbles between the metal ions in the metal, and therefore it sinks to the bottom. Also, quite interestingly, these are nice little experiments you can do at home. One thing about air bubbles also depends on how tightly packed the molecules are. So that can also mean how much of a molecule is dissolved in the solution. So for example, if you took some household sugar and you dissolved it in water till no more sugar will dissolve, and then you take the liquid, so you have a saturated solution, and then sort of serially dilute it, so you have lots of different solutions getting more and more dilute, and then you coloured them all. That's what these are here. And here we have, so going from the least dense here, so the most dilute, to the least dilute, the most concentrated here. And then you can actually layer them on top of each other. So this is different sugar layered solutions. And the reason that they layer is because of the different densities. They don't then mix and separate. Also, here on the right hand side, I've got a rainbow of sort of familiar liquids. So at the top, we've got olive oil, which runs into vegetable oil at the bottom here. And then we have wine, which runs into water. You can see that water is miscible with wine, as you've got sort of the red layer of the red wine and the blue layer of the water, and in the middle you've got a bit of purple. And then here we have dish soap, and then right at the bottom with a really high density we've got maple syrup. So that's quite a nice experiment for density as well that you can do. So that's sort of it for our vinaigrette. We're going to now listen to a bit of a chat from Jonas Lodge, who will introduce himself in a second via video link, who's going to show us how you would make mayonnaise and vinaigrette. Before we do that though, we use this concept in the lab, I use this every day. We use it to separate our compounds. So as you can see, we have our organic or our oily layer on the top here, and we have our water layer on the bottom here. And what I can do is, if I think about it, I can make molecules or my compounds move between the two layers depending on where I want them to be. And then at the end, I make them all go into the organic layer. And then that's how I extract my compounds from my reactions. On the left-hand side, the organic layer is on the bottom. This is because this is a different solvent. This is actually a much heavier solvent. So it has a much higher density, and these are sort of chlorinated, so things like chloroform. But back to Jonas, who's going to now talk about making a vinaigrette and a mayonnaise. Hopefully he works.
Hello, my name is Jonas Lodge, and I am the owner and head chef of restaurant GL50 in Cheltenham. So first of all, we are going to do a vinaigrette. This is going to be a blueberry vinaigrette made with our own blueberry vinegar here. So a good vinaigrette is made with a ratio of 70-30, so 30% vinegar, whichever vinegar or acid you fancy, to 70 oil. This is just regular vegetable oil. I'm going to use a hand blender for this one because after the mayonnaise I'm going to be tired. And you're going to slowly pour this in. Oh, it smells amazing. Slowly add it, you can see it starts to thicken up. Starting to get really thick now, you can hear it, you can't quite see it. So it's starting to coat the end. We'll just keep going a little bit longer. You can see this is nicely emulsified. So we'll leave that there, a nice pink vinaigrette. Now what we're going to do is we're going to leave this here whilst we are making our mayonnaise. We'll come back to this in a minute. So the mayonnaise, I've got 3 whole egg yolks. I've got a teaspoon of English mustard. We're going to add a little tiny bit of our own apple cider vinegar. A little bit of acidity, you could use lemon juice as well. And then this one we're going to do by hand. Hopefully I won't hit this vinaigrette. Just use a tea towel to stable it. Tricks of the trade. And then the same as the vinaigrette, we're just going to slowly add oil and keep whisking. See, it's getting nice and thick now. Slightly harder to whisk with one hand. Just try that. There we go, so that's a nice thick mayonnaise. And to finish this, we should add a little bit of salt, get a little bit more acid, very important with all the grease that's been added, or lemon juice you could use. Nothing like the colour of real mayonnaise. we go. And as you can see, hopefully on there, nice, thick, emulsified oil and vinegar. And so now we are going to pop back to the vinaigrette, which hopefully, as you can see on here, is already starting to separate. So it's no longer thick, and you're starting to be able to see how that oil and vinegar are no longer staying together. See, it's no longer a thick, completely opaque liquid, and on my spoon you're left with some of the oil and the vinegar as two separate things. And that is in a short space of time. As I've been making that mayonnaise, you can already see that this doesn't want to be together anymore. And eventually this will just completely split. Thank you for watching.
As Jonas just showed us, we have the vinaigrette which separates, and we also have the mayonnaise which is stabilized. So how do we stabilize the mixture? So we either break up the globules, so we have distance, so space separation is less likely, or we add a stabilizing agent. And doing one or both of these things leads to the formation of a colloid. I've got the definition of a colloid here. So a colloid is a mixture where one substance of microscopically dispersed insoluble particles are suspended throughout another substance. So what that means when I say insoluble, it means they don't necessarily want to mix. So it's like the oil and water. And as you can see in the top right hand corner of this slide, you've got lots of little globules suspended in some other darker medium. There's more than one different type of colloid. So I've got some here. These are mostly food related, but I struggled with a few. So solid gas, for example, styrofoam. So that's gas dispersed within a solid. Liquid gas, so this is whipped cream. Liquid, which is an emulsion, which is our mayonnaise or milk, liquid, or liquid solid gives us sort of jelly texture. So how do we actually stabilize the oil and water or oil, water, lemon juice, vinegar mixture in mayonnaise? And the reason that this is stable is because we've added egg yolk. So egg yolk contains both water, but also contains emulsifying agents, which go by the collective name lectin, which I've written here. So lectins are amphiphilic molecules, which we talked about earlier. So they've got a polar or hydrophilic head group, and they've got some very hydrophobic tails. So they've got both characteristics in one molecule. So there's lots of different types of these molecules within the sort of group of lectins. So some of them have sort of modified phosphate heads compared to the phospholipids we talked about earlier. So here's one that has a choline group added, an ethanolamine group added, an inositol group added. This is an amino acid, so the things that used to make up proteins added here. You can see that it's charged in this form, so this means it's very hydrophilic. And there's also the type we talked about earlier, which is just a sort of naked phosphate at the top being our hydrophilic head. So from now, I've sort of highlighted how this relates to the structure here with the sort of my blue dots, purple dots, and pink tail. But from now on, I'm going to draw them as this sort of simplified version where we've got a hydrophilic head group here, and then a fatty acid tail, which is hydrophobic here. And what happens in our mayonnaise is we add these amphiphilic agents or the surfactants to our water oil mixture, and they give us a colloid because they stabilize the oil droplets in the water, because the hydrophobic tail sticks into the oil droplets and the hydrophilic head sticks into the water. And therefore, because you've done that, you haven't got the really unfavorable interactions between the oil and the water molecules, because you've now got favorable interactions between the oil and the tails and the water and the heads of these amphiphilic molecules. So lots of stabilized droplets prevent the separation into two layers. So that's why we have a colloid and why mayonnaise doesn't phase separate. Okay, so moving on a little bit more to the sort of key aspects of our pizza. We're going to talk about the dough, which we've made. So dough and flour contain gluten. Now, gluten is a family of proteins which are found in grains. And when flour is mixed with water and beaten, gluten, which is a water-insoluble protein, the proteins stick together to form crosslinks. So as you can see on the left-hand side, this is a bit of dough that's been worked a little bit. So you can see that there's some crosslinks between chains, but there's still some pretty big holes. And then on the right-hand side is a piece of dough that's been worked a lot more. And you can see there's some very, very strong links between the chains. And what this does is it forms a big elastic network of cross-linked proteins. And it means that we get air bubbles, which can expand within our dough, but they don't burst. And gluten also is important in providing a satisfying chewy texture to our dough. So I'm going to talk here. This is, I think, Jonas's pastry chef making us some dough and showing us how elastic the network is. So this dough has been worked and now they're stretching it out. And you can see that if you hold it up to the light, it's actually so elastic that you can actually stretch it out and see light through it. And that's called a window pane test. And it's very common in bread baking. If you watch the Great British Bake Off, they will have talked about that non-stop in Bread Week, I guarantee you. But I've just mentioned that this elastic network prevents the air bubbles from bursting. But where do air bubbles come from? So it comes from the fact that we generally add yeast to our bread dough. So what yeast is a microorganism which can come in two forms, either fresh form here, which is generally used in restaurants, and dried form here, which is generally what you would buy in the supermarket. And the dried form is sort of revitalized when it's mixed with water and it restarts its metabolism. And what the yeast does is it feeds on the sugars in the dough and it respires and makes gas bubbles. So respiration is sort of the process by which they get their energy, but they also, while they do that, they release gas bubbles, which are trapped within our gluten network, and this inflates the dough. So at first, we have plenty of oxygen within our dough, so they can respire aerobically, the yeast particles. And then when we run out of oxygen, this process becomes less efficient. So you can see here, one sugar molecule, one glucose gives us 6 carbon dioxide molecules. And then when we run out of oxygen, the yeast has to respire anaerobically. So one glucose molecule is only converted to 2 carbon dioxides, but nevertheless, this still happens. So these gas bubbles inflate our dough. And then when we bake it, the heat causes the gas bubbles to expand even more, which is why we get very big bubbles in our bread. And eventually, with the heat of the oven, the yeast will die because the heat causes the proteins, the enzymes, the workhorses that actually do this process to unravel. So as I said before, proteins, their function is very closely linked to their structure. So if they unravel, they completely lose function and basically the yeast dies. And this process of the proteins unraveling is called denaturation. So here I have another little video. Hopefully that one's gonna, no, come on, you can work. No, that's going to work. Let's try it. No, it's not. There we go. So you can see that as we prove our bread, we normally do this in sort of a warm room. The yeast respires, and that causes the release of gas bubbles, so carbon dioxide bubbles, which are then inflating our dough, and you can see them all down the side of this bowl here. So we've now made our dough for our pizza, but what's the most fun bit of pizza is toppings. So we're making a vegetable supreme pizza today, so we really want to have lots of colourful and variety of colourful vegetables on the top. And where does this sort of diversity in colour in vegetables come from? What are the chemical roots of this? So to start off with, I'm going to just explain how colour works. So the physics, the basic physics of it, I'm not a physicist, so this is going to be quite straightforward physics, in that white light is made up of lots of different wavelengths of light. which gives us the visible spectrum. So wavelengths are, light is a wave, and wavelengths are from the sort of top of one to the top of the other, or the middle of one to the middle of the next, the same point in the next wave, and that's the wavelength. So all of the wavelengths that make up visible light also make up the sort of colours of the rainbow. So we have red light, which has the longest wavelength and is the lowest in energy, all the way through to violet light, which has the shortest wavelength and is the highest in energy. And this forms part of the electromagnetic spectrum. So you can see here that this tails off with red to infrared, which is basically heat energy, and ultraviolet is the violet end, tails off into that, and we go up to sort of X-rays and gamma rays as you get higher and higher in energy. So how does this work with colours and what we see? So white light is made up of these wavelengths, and the absorption of some of these wavelengths means that others are reflected, and that's the colour that we see. So you can see here I've got a carrot in the middle of my screen. So if we shine white light on the carrot, we can see that the carotenoids in the bottom, these are pigments within the carrot, they absorb in the sort of violet to green region of the spectrum. So what is reflected is what is not absorbed, and therefore we see the sort of yellow, orangey, red end of the spectrum, which is why we see the orange carrot. But what's the chemical root of this? What is actually doing the absorbing of the light? So it's generally an organic molecule, and it's generally organic molecules that can absorb light and visible wavelengths of light are what we call conjugated, or have conjugation within them. What this means is there's alternating double bonds and single bonds as we go down the structure. We can also include some heteroatoms, so that's normally an oxygen or occasionally a nitrogen in there. But generally it's alternating double and single bonds. And what it means is we've got sort of free electrons in here. Don't worry too much about electrons if you've never heard of them. They're just a part of the atom. So what we have here is a carotenoid. So beta carotene is a carotenoid, and there is our conjugated system. And so this is what absorbs at the violet to green end of the spectrum, and therefore what we see is orange. So popping that on our visible spectrum, we can see we've now know what gives us orange light, orange coloured vegetables. But most vegetables are green, right? So what's going on there? So what is sort of very important is we've got chlorophyll. Chlorophyll is the pigment that's responsible for plants' green colours. It's a key pigment in absorbing light for photosynthesis, which is how plants effectively make their own food. And it lives, chlorophyll resides in chloroplasts in cells, which because of the chlorophyll are also green. And there's two types, chlorophyll A and chlorophyll B. These are the conjugated systems. So I've said we can have nitrogens in there, we can have oxygens in there. And the exact sort of nature of this conjugated system really has an impact on the wavelengths that are absorbed, and therefore the wavelengths that are reflected and we see. So you can see here there's not much difference. It's not helped by the fact that I've got one of them upside down relative to the other one, but the only difference I can really spot is we've got an extra group here. So we've got this added group, this aldehyde in chlorophyll B that we don't have in chlorophyll A, and that is enough to change the absorption spectrum. So you can see that chlorophyll B absorbs a lot more in the blue region of the spectrum. So therefore the colour of that is a lot brighter green, whereas chlorophyll A, you can see it doesn't absorb quite so much in the blue region as chlorophyll B. Therefore, we sort of see more of those blue wavelengths coming through. So the colour green that we see is a bit deeper, remembering that the colours that aren't absorbed are the colours that we see. So if we pop those onto our spectrum, I've just popped one on for space, but there's our green. Now, sometimes the colour observed depends on the ripeness of the vegetable or the fruit. I found this out when I was doing some research for this session, but some fruit and veg will only ripen on the plant, whereas others will ripen once you've picked them, and you can use ethene gas to do that. An example of those are tomatoes or bananas. But peppers are an example of a veg and fruit which only ripen on the plant. But what causes this dramatic colour change? So red and orange and yellow peppers all originally start life as a green pepper. So we know what causes the colour to be green in green peppers, it's chlorophyll. But what happens is as we go through the ripening process, the chlorophyll decomposes to give us other compounds. So here we have some. We have lutein, violaxanthin, and our friend from our carrot, beta-carotene. So here we have different conjugated systems. You can see we've still got our alternating double and single bonds. And it's actually the sort of the specific nature and even length of our conjugated system. So you can see here, this is the shortest one in violaxanthin. And this is therefore the brightest yellow that we would see, the brightest yellow pigment. It's a little bit longer in lutein. Here we've got an extra bond added on the end. So this one is again a different color, and this one is a sort of more towards orangey yellow, more of a sunshine yellow. And then again, beta carotene, we've got another bond in conjugation. We've got a longer conjugated system. The nature of the conjugated system is different, and therefore that's why we have a different absorption spectra. So what we see reflected into our eyes is different. And then going even further to the red pepper, we get heteroatoms, so oxygen incorporated. So you can see here we've added, this is a ketone, so we've added a double bond to an oxygen here. And this changes the colour again because we've changed the nature of our conjugated system, and that's why it's red now. And then you can see if we add another ketone group, so we add another heteroatom in this position, that changes it again, and we get a sort of deeper red. This is more sort of orangey red in capsanthin, and capsorubin's more of a deeper ruby red. So it's interesting that we can see here how the exact nature of that conjugated system is deeply affecting the colour that we observe within our peppers. So they are now on our spectrum. Okay, we're going to just sort of have a little bit of an interlude. Sometimes the exact nature of a chemical species changes depending on the environment. And this is because the chemical species exact nature is slightly different. But what do I mean by a chemical environment? I mean the sort of chemical surroundings. An example of environment would be the pH of the solution. Now, pH is a measure of how acidic or basic a solution is. So acids have a relative excess of H plus or hydrogen ions compared to hydroxide ions, and vice versa for alkalis or bases. They have a relative excess of hydroxide ions compared to hydrogen ions. And we measure how acidic or basic something is on the pH scale, which is shown for you here. So at the sort of top end of the scale at pH 0, we have lab acids that I deal with. And then we can move a bit further down. So pH 2 is about lemon juice, pH 3 vinegar, pH 6 we're getting to milk, 7 neutral, that's water. If we get to pH 9, that's baking soda or bicarbonate of soda, they're the same thing. 10 is antacid tablets, and then if we get to sort of 12, 13, 14, we're in bleach or drained tick cleaner territory. So if I'm in the lab, how do I tell what pH something is? Now, we can either use a fancy bit of kit called a pH probe to give us an exact value, but I don't really need that in my lab. I just need to know, is it acidic or is it basic or alkaline? That's all I generally need to know. So I use an indicator. Indicators are chemicals which change colour depending on the pH of the solution. So in the lab, we use indicators. So this is universal indicators, a universal indicator paper here. So this changes colour, and it's a mix of different indicators to change colour to tell me what pH I'm at across the whole of the pH scale. We also use these titration indicators. These are more common sort of at school, A-level chemistry, sometimes at GCSE. So this is phenolphthalein. This is colourless in acid and bright pink in alkali. And similarly, bromothymol blue has a bit of a smaller pH window. So below about pH 6, it's yellow, and above about pH 7.5, pH 8, it's blue. And it also, interestingly, in very concentrated acid, apparently it's hot pink. I've never used it, so I wouldn't know. But some foodstuffs have compounds which also can act as indicators. An example of this is red cabbage, which has anthocyanin pigments. And at different pHs, these pigments can lose or gain protons or H+ ions, and this gives us different species. And because we have a different species, it has a different conjugated system or a different nature to the conjugated system, and therefore it has a different colour. So this is where I'm going to undo my video, hopefully. Put my video back on because I've got some of this indicator to show you. Okay, here we are. So at pH 7, so that's neutral pH. I'll get my water out. So there's just tap water. And then here I have some of my indicator. It's a lovely purpley colour. So hopefully it's the same colour I promised as you see in the bowl on the thing. So you've got the sort of bluey purple colour there. So that's pH 7. If I add acid to that, so that's our blue colour. If I add acid to that, I increase the concentration of H+ ions. And what happens, we've got our blue at the top, that's the same species that we've got in our glass here, is that we can pick up a H+ ion, I need my laser pointer back, at this position here. So that's the only difference between this molecule here and this molecule here is we've picked up H+ here. So what that does is it changes the nature of our conjugated system, which means we've got a change in colour. So I'm going to add some vinegar to this. Hopefully it's not too acidic, or we will be able to stop a little way. So you hopefully can see that's gone sort of pinkish. And then if we keep adding acid, and I'm not sure I'll get that concentrated with vinegar, but we can give it a go. It now smells like a fish and chip shop in my room. It's lovely. We can actually pick up another proton at this position because this oxygen atom here helps out. Don't worry too much about how this works. But we basically pick up another proton, change the nature of the conjugated system yet again, and therefore change the colour, and it goes a brighter pink, which maybe we'll get, maybe we won't. Not really. Maybe you can see that, slightly brighter. If I had some proper concentrated acid, we could get it to go basically red, but I don't really have that lying around in my house. Okay, so that's acid. What happens when we add base? So I'm going to get myself another glass. And here we go. I'm going to add some indicator to it, and we've got our blue colour again. And then I'm going to add some bicarbonate of soda, or sodium bicarbonate, or baking soda to this. I'm going to add quite a lot, but you know, we only live once. Hopefully you can see it's kind of gone a bit more greeny blue, or a deeper blue. It's proper blue now. It was sort of bluey purple, and now it's a proper blue. Hopefully you can see that. So what happens there is, as we add base, this is quite a mild base, so it doesn't give us a massive colour change. We reduce the concentration of H+ compared to hydroxide ions. So effectively we increase the concentration of hydroxide ions. So what happens, and we haven't really got strong enough base to do this, but why it is going slightly more bluey green, you can actually see it is actually going a bit greener. I don't know if you can see in the camera. The relative excess of hydroxide will basically attack this molecule, and it attacks at this position here where my laser pointer is, to give us this molecule here. That's the difference between those two, and it's effectively a greeny colour, which is the mixture between the green and the blue. You can see here. For a bit of fun, you can see that this indicator does change, and I'm not going to use this white wine vinegar, so we might as well chuck it in. So this is our basic solution. So we've chucked it in with this. Hopefully I'm not going to add too much, and I'm not going to do it with a volcano. There you are. You can now see it's going through purple. I'm getting a volcano. Through purple and into sort of, if we keep going, I think I'll run out of space in the glass first. You can see as I add it, I get sort of a plume. I'm going through purple anyway. I think I will run out of space in my glass or overflow all over my desk before I get there. But you can see that it does change colour based on the concentration of acid or base in our solution. I'm going to move those away before I knock them over. Okay, so we've done indicators and foods. So From there, we can see that we have got different compounds which have different colour, but this is all to do with differences in the conjugated system within the molecule. So we've got our beta carotene and sort of other chlorophyll decomposition products that go through with our peppers. And then we've got our chemical species, which is the same compound effectively, but in different environments. The species are different, and that changes the nature of the conjugated system, so we have a different colour. The basic take home from all of this is that the colour of organic compounds or species is dictated by the precise nature of the conjugated system, and this allows us to have a sort of lovely rainbow created by chemistry. Now, I'm just going to switch gears a little bit, and we're going to sort of leave our pizza alone for until we get to questions at the end. But I've always been fascinated by how molecules can interact with our body systems to have sort of bad side effects or fatal side effects. I've always found the science of how poisons kill people really interesting. Thankfully, or hopefully, our food is not laced with poison, but there are some really interesting compounds that could give us harmful side effects that are found in our food. Should we be worried about these? So we're going to have a bit of a myth busting section here. So it's a bit of a common myth that apple seeds contain cyanide, and therefore eating an apple seed is really bad for you because cyanide is a really toxic compound. There is actually a grain of truth to this in that apple seeds do contain a compound called amygdalin. Now, amygdalin, in this form, is absolutely fine, quite innocuous. There's 2 sugars on here, and then this little group added on the end. But what happens is if you finally chew an apple seed and amygdalin is released into your body, you metabolize it. And so the enzymes in your body break it down. So you take off that 1 sugar with the first beta-glucosidase enzyme, then it takes off another sugar, and then you end up with this mandelanitrile group. Now, this compound here is unstable and decomposes to give us benzaldehyde, which is fine. This is not a problem at all for our system, but also hydrogen cyanide. Now, this is a problem. This is highly toxic. You need about 1.5 to 2 milligrams per kilogram of your body weight to get a lethal dose of this. So that's not very much in comparison. The sort of typical paracetamol dose for a sort of average 75 kilo adult is 13.5 milligrams per kilogram. So not very much at all is needed to sort of kill you if you've got with hydrogen cyanide. So should we be worried about eating apple seeds? Really, realistically, the answer is no. There's only 3 milligrams per gram of amygdalin within apple seeds. Each seed's about 0.7 of a gram. And amygdalin's only released into the body if the seed is damaged. And even then, not all of it is converted into hydrogen cyanide. You'd have to eat about 85 apple seeds. And when I say eat, I mean finely chew and swallow about 85 apple seeds to get any form of adverse effects. And to actually achieve a fatal dose, you would have to eat about 200, or at least 200. And again, that's finely chewing them and swallowing them. So in summary, apple seeds do not contain cyanide, but they do contain a compound which can release cyanide in your body in small amounts. This isn't exclusive to apple seeds at all. There's other fruit seeds and stones. So here's the amygdalin content here of other common fruit. So we can see that some of the stone fruits like apricots, peaches and greengage have quite a high amygdalin content, and cherries, pears and nectarines are much lower, a bit more comparable to our apple. But again, if the seed or the stone isn't damaged and it passes through the body whole, there's no problem with this at all. You would have to sort of finely chew and eat these seeds. I mean, good luck to you if you can finely chew and eat an apricot stone. But generally, these fruits are fine. If you swallow the odd apple seed, you are fine. So moving on from this, which was kind of a bit of a myth, has anybody, I don't know if you can use the raise hand function at all, if not, no worries. Has anybody heard that it can be harmful to drink grapefruit juice with certain medication? I don't know if I'm getting hands or not. Okay, well, for some people that take medication, it shouldn't be mixed with grapefruit juice, which sounds a bit strange, but what's actually going on here? Well, it's all to do with the way that we clear drugs from our body. So in a healthy person, well, not a healthy person, a person who has to take one of these drugs, who's not taking grapefruit juice with it, or drinking grapefruit juice, or eating grapefruit, they take the drugs go into the body, they have their desired effect, they help to relieve the symptoms of whatever the problem is, and then they go and they are cleared by these enzymes called P450 enzymes, but basically they're the clearance enzymes of the body, and then that means that it's excreted, the drug molecule, and then you have your healthy person who's had their sort of whatever their problem was they were taking the drug for has been resolved. Now, what happens when we mix that with grapefruit juice? Well, grapefruit juice contains certain chemicals, bergamottin and naranjin here, and these chemicals here, dihydroxybergamottin and epoxybergamottin, these are metabolites, so this is what this can turn into in your body. And similarly in the body, similar to the amygdalin, we can chop off the two sugars to give us naringenin here. And these compounds here, and quite high concentrations in grapefruit juice, and they are inhibitory, they inhibit the clearance enzymes. So this enzyme here doesn't work properly if you've got lots of these molecules hanging around. So what that means is if you happen to take grapefruit juice in combination with such tablets, you inhibit the clearance enzyme, which basically means that the drug builds up within your body, and it gives you harmful effects, or eventually leading to overdose. There's some suggestion that this could be sort of a beneficial effect in some drugs in some cases. There's one paper about this that I've seen where they've suggested this could be useful, this grapefruit juice effect, for anti-AIDS medication, which would otherwise be broken down really quickly by the body by the P450 enzymes. So the idea is, do we inhibit the P450 enzymes with the grapefruit juice? And therefore the drugs have a sort of better lifetime in the body. As a scientist, I'm not quite sure how this would work, because there's quite a lot of variables there. How much grapefruit juice do you drink? What do you eat? How much do you absorb? How big is the person? And I think the only way they could possibly get away with doing this, or be able to do this, would be to put the key compounds, the bergamottin and the naranjin, into the drug, sort of the tablet and the formulation itself. It's not anything to be worried about, this grapefruit juice effect, really. If you're worried about medication that you're on, it's always in the leaflet that comes with the medication saying, do not take with alcohol. If it's a problem and a known grapefruit juice interaction, it will say, do not take with alcohol and grapefruit juice, or grapefruit juice. And if you are at all concerned, contact your GP. I don't have an exhaustive list of the drugs that this affects. So if you are at all unsure, please contact your GP about that. So hope you guys have enjoyed this sort of chat from me about this. So if you're interested in the science of cooking and you want to know some more, here's some books that I've used a lot in making these talks. So on the left-hand side here, we've got Why Does Asparagus Make Your Wee Smell? Which has a strange title, but is a really good book. It's all done in a sort of, each chapter has a little question, and the chapter goes through to answer the question. Each chapter is very, very short, about 2 pages. And it tells you all sorts of things like, why does garlic make your breath smell, and things like that. Very, very interesting book. There's also the Compound Interest blog, which does contain some of the same stuff as this book, but also has the science, not just food science. So it's got what's it got? It's got oil science, it's got paint science, it's got probably cosmetic science. Also, I noticed recently the Nobel Prize announcement came out last week, and they had a lovely blog post explaining for sort of a non-chemist what this prize was about, and what the work was actually that had been done by Ben List and David Macmillan to sort of be worthy of this Nobel Prize. So that's worth checking out if you're at all interested in chemistry, but don't sort of have an undergraduate degree like myself in it. Also, slightly less hardcore science would be these Dr. Stuart Farramon's books. So these have a lot of explanations in them. They have less chemical structures in them, what I've been talking about today. The Science of Cooking is very much a pop science book explaining how the cooking process works. The Science of Spice is probably closer to a cookery book. It explains what spice goes with what and why. And the Science of Cooking here, Peter Barham's book, is probably somewhere between the two in terms of how much science is in there, how much sort of popular science is in there, etc., etc. In terms of other sort of outreach events linked to Oxford University, my department, the Department of Chemistry, do run some outreach events, mainly through schools, but definitely worth getting in touch with them if you fancied having a go. They had something on their website when I had a quick check about chirality and COVID, which sounded quite interesting. And then under that, I've dropped a link for Oxwe Chem. This is a sort of relatively new student society called Oxford Women in Chemistry. I happen to be on the committee. They've just started up an outreach sort of program. They're just getting going, but they're really, really keen to get more women into science, especially into chemistry, and hopefully get more women in to do PhDs and join us, because we're great and we need more of us. Also, not related sort of as such to the university, but obviously please check out all of the ideas festivals, great events that they've got going on throughout this week, next week, and in future years to come. And with that, we get to my thanks. So I'd like to thank Jonas from GL50. He was going to try and be here today, but has had to go off to a vineyard to do some wine tasting in Hungary. But thanks to Jonas for doing those videos for us. Thank you to the Royal Society of Chemistry for funding this event and supporting it. Thank you to the team at the Oxford Science and Ideas Festival, Dane and Kathy, for your vision with this event, your help and support in making it happen. And I have to thank my funding bodies and my university for sort of letting me be here. So SBM is my course that I'm on, and how I am funded to do my PhD. And with that, I guess I'm going to open the floor to questions, and I think Dane's going to take control. So yeah, any questions about the chemistry confections, any questions about chemistry in general, what it's like to do a PhD, etc., ask away.
Great, Jess. Thanks so much. I hope everyone enjoyed that. I certainly did. And we've got some questions that have come up in the chat box. And if anyone would like to drop some questions or some thoughts through, I thought it might be also interesting. One of the things that we could do is almost kind of put together a word cloud inspired by your grapefruit kind of word cloud. Just any thoughts that come to mind when you think of chemistry. So oftentimes people will think of kind of petrochemicals and things, but any kind of ideas that kind of come to mind when you think of chemistry, if you just drop them in the chat, that might be quite fun just to see what people are thinking about at the moment. There was one question about vegan lectins, I think. So when we were talking about the emulsification, when Jonas was doing the vinaigrette, obviously he had an egg, and it was the egg that stabilised the boundary between the oil and the water layer. And obviously some people might not want to have animal-based foodstuffs. So I just wondered if there are any thoughts on vegan lectins. Maybe it comes from peas or beans, something like that, a protein.
I mean, possibly. I mean, these types of molecules are prevalent everywhere. There will be plant-based versions. I don't know exactly where you'll find them, but the same kind of molecules will exist within plants. Definitely the phospholipid type ones, because they form cell membranes, so they're everywhere.
Great. So I think one of the key things that a sort of emulsifying or a stabilizing agent does is to just try and control the way the particles in a solution can kind of group together. So obviously, if you've got an oil phase and a water phase, when you mix it all up and there are fine suspension of oil in water, or even the other way around, if you've got mostly water in oil, it's the stabilizing the interface between these two different phases, the oil phase and the water phase. That's what we're trying to achieve with an emulsifying agent. So I think even if you have even something like chalk, can help. Obviously, we don't want to eat chalk in food, but something that stabilizes the interface between the two different phases is going to help control the way the colloid or the emulsion kind of maintains its structure. So it doesn't have to be a detergent that we might be familiar with washing our dishes. So just kitchen soap is one kind of way to sort of stabilize mixing between different phases, but even kind of eggs. And I think that I had a quick look, and soybeans is one route for a plant-based emulsifying agent, but even sort of mustard and all sorts of different things that have these different properties of having a water-liking end and an oil-liking end. If that can sit at the interface between the oil and the water kind of phases, or even if it's just a non-polarized kind of solid, like a really kind of fine grain, if that can just be at the boundary, I think that can stabilize things. Obviously, we're not suggesting that people add chalk to their food, because that's not going to be very healthy. There was a comment about the peppers. There was quite a bit of discussion, I noticed, around the different colours of peppers, and someone was surprised to find out that green peppers are just unripe red peppers.
Yeah, no, that surprised me as well. I don't really like peppers that much, so I never really thought about it. And then I looked, sorry, I'm trying to get my mouse back so that I can see the chat as well. So yes, indeed. So because of peppers, I was fascinated, but I found that out and I thought, well, why does that make such a difference to colour? And it is this weird sort of decomposition. And as a chemist, I can sort of track the way that it's doing it, and it's possibly even more interesting to me as well, because I can see what's going on in the compounds.
So as we're talking about making a vegetable supreme pizza, we thought it'd be a good idea to roast some carrots, add a bit of salt, a bit of oil, and put them together with some other brightly coloured fruits and vegetables, some yellow tomatoes and some courgettes, and just think about different coloured fruits and vegetables, but particularly carrots. So Jess, what do you think about all these different sorts of coloured carrots that we can get today?
So there's awful lot of different colours of carrots. They're sort of back in, we'll say, ye olden times. There was lots of different colours of carrots, not just orange carrots, purple carrots, there are sort of yellow carrots, sort of white carrots. And the reason why these are all different colours is just to do with the pigment that's involved and the concentrations of the pigments. So in purple carrots, they have the anthocyanin pigments, which is the same sort of family of pigments that the red cabbage has, which gives us those sort of funny indicators. And then over time, the orange carrot was popularized, and then that's why we normally just see orange carrots. But the sort of other colours of carrots are sort of creeping back in as a bit more like fancy food, I would say. I guess Jonas probably uses them in his lovely restaurant in Cheltenham quite a lot. But yeah, different coloured carrots are all to do with the different pigments that are in the actual carrot.
And obviously the concentration of the different kind of pigments changes what the carrot looks like. So if there's a lot of kind of the kind of orange carotene in there, but if there are different molecules in there, that will shift the spectrum.
And the carrots have sort of been bred together, and we've been only really used the orange was favoured, so that's the reason most people have produced the orange ones, and then the others have just sort of faded into the background, but still sometimes used in cooking.
Great. There was someone suggested in the chat here, there's agar maybe an option. I'm not sure about that. I'm
not sure how agar works. I think it's more of a sort of colloid situation than I assume this was as a suggestion for a stabilizing agent or an amphiphilic kind of molecule. I think when I was looking at the colloid stuff, I think it is actually a colloid because it's a bit like a jelly. So I think it's more of a solid liquid colloid than a thing, but possibly, I'm not entirely sure.
That's something to look into maybe for next time. We'll maybe get back to you about that. Oh, and flaxseed might be an aquafaba, of course. Yes, from the chickpea, the can of chickpea juice as one option. So I don't know if you've had any more thoughts on different colours of foods. One of the things that we were going to talk about was the different types of dough that people might make. So thinking about yeast and sourdough. So we printed in the programme an option for making dough using yeast. But there's, in the lockdown, I think there was a bit of a revival of making sourdough. And I don't know if anyone in the session today has experimented with making sourdough and exploring the flavours, the different depths of flavours that comes out of it. I don't know if you've ever tried making sourdough bread, Jess, yourself, or if it's just one step too far.
I've made the cheats version where you use yeast and you leave it overnight to capture a bit more yeast from the air, but I've not got the patience to make my starter for weeks on end. I'm too hungry for bread. But the difference between sourdough and sort of a yeasted, that you would buy the yeast dough, is to do with where the yeast comes from. So when you make a sourdough starter, you mix the water and the flour, and then you leave it sort of fairly open to the air, and that captures sort of yeast that's just kind of floating in the air, and then it sort of develops and reproduces in the starter, and you leave it. I think you need about 2 weeks to get a half decent starter. And you have to keep chucking bits away. It seems like very much of a faff to me, but you know, it does give you nice bread, I'll give you that. And that basically develops the flavour as you go. And the sourness, I believe, will come from that anaerobic respiration that we talked about. It makes ethanol, which is basically alcohol, the same alcohol that would be in wine or whatever. But then that is broken down further into lactic acid, which is probably why sourdough is sour.
It's a slightly acidic flavour, almost like the kind of lemony, but not quite as acidic as the sort of lemon.
Yeah.
And I suppose if you have more of these, if you kind of leave your dough to kind of prove or develop over weeks and months, or even the whole 18 months of the lockdown when people have been experimenting with making sourdough, I suppose the range of molecules that are in your mixture might increase the kind of the taste spectrum. Would that be fair to say? Yeah,
so the more different compounds we've got, the more sort of the broader the spectrum, the different combinations will give us different flavors, and broaden sort of the flavor profile as you go through. And then that's to do with the sort of diversity of compounds that you will form from the yeast in your starter, and respiring and acting and reactions happening as you go through.
All right, so if people have that time on their hands, they can always experiment with developing that flavour profile of their sourdough. One of the things we were also thinking about was, obviously, we were thinking about pizza. And all the various different toppings that can go on it. So maybe vegetables. Someone put a comment in about they like meat in their food, which is obviously great too. But sometimes people might want to put some cheese on a pizza.
Yeah.
And we had a conversation a little while ago about why does some cheese go stringy?
So that's to do with the exact kind of makeup of the cheese. So the cheese is basically, there's lots of fat and there's protein as well, and it's to do with the amount of fat between the protein. Cheese is basically a colloid, it's a big mixture, but it becomes quite colloidal, which is why we get the stringiness. And if you've got the right ratio of fat to protein, you can basically stretch out all of those chains and you get big, long, stretchy, stringy cheese. So it's to do with the exact makeup of the cheese, and that's why some cheeses like mozzarella are very, very stringy, whereas some cheeses like a Stilton or sometimes a cheddar tend to like burn and not string as much and just tend to sort of blob, if that makes sense.
Yeah, gloopy. I suppose we had the video before where Jonas was making it's the protein molecule, so the glutinous protein molecules, isn't it, where the window pane test is stretching out. I suppose the the molecules, if they're able to kind of cohere together and become thinner and thinner and still hold themselves in a shape, like the window pane test for pizza dough, or the kind of stringy cheese that we like with the mozzarella kind of effect. It's the same chemical processes, would you say?
Yeah, I think so. Yes, same sort of idea in that you've got the stretchiness coming from those protein chains.
And someone's dropped in the chat, I think pizza is cheese on a pizza is for the umami, so the sort of savoury flavour. So we haven't maybe mentioned the umami today, I don't think, but the kind of that sense of flavour of just richness. And so, do we know why the umami flavour is so popular? Things like that kind of marmitey kind of taste.
I'm not sure what the sort of chemical background is to that. I imagine it's very, very complicated. I don't think it's something like, sweetness is very straightforward. We know why things are sweet. We know why things are sour. I think bitter is very complicated, and I feel like umami is going to be very, very complicated as well. I feel like there'll be a massive range of compounds that give you that kind of flavour, and it might even be mixtures of compounds.
It might even, I would imagine it also might be kind of mixed up in the smell. So obviously we kind of smell some things and we taste some things. The mixture of compounds that you're talking about, I suppose there are some really small molecules. So you had your molecular models of ethanol, the small, the black and white kind of plastic models. Molecules of different sizes might behave differently in the room around them.
Yeah.
Right. So if you maybe held up your ethanol molecule and your larger molecule that you've got made out of monomers, just so people can see, Jess.
Yeah. It's not ethanol; it's actually acetic acid. But
acetic acid. Sorry.
I can turn it into ethanol. Imagine two hydrogens are here and here. That's ethanol.
But a pretty small molecule like that.
To me, this is tiny. This is quite, I work on small molecule chemistry, and this is still quite small. We would class large molecules as proteins, which could have thousands and thousands of, there would be thousands of Daltons. So that's hundreds and hundreds of atoms joined together. And we don't even do the skeletal notation for that, because that's too complicated for that. So then we have more notation. But yeah, hello. So yeah, different sizes of molecules will interact differently. Things that are smaller tend to have more of a smell. They're more likely to sort of, they're what we call volatile. They're more likely to waft up your nose. Whereas bigger molecules, much less likely. This is menthol, so we know what menthol smells like. This is still small enough to waft up our nose and be around in the atmosphere. But yes, different sizes of molecules will, and which receptors they interact with, will have massive, massive impact on flavour and taste.
So again, just that side, the sort of the chemistry of the substance. So if it's a small thing, it might float around and go up your nose. If it's a huge thing like a protein, it might be a gloopy structural kind of molecule. But I don't know if we've got to the bottom of the umami thing, but I think, so looking at this book that I've got, it's another one of these sort of books, so it's Science in Cooking, and there are some notes saying that the umami flavour is a potentially high energy food, so it might be a really kind of, again, a rich, a bit like the sourdough, a really rich flavour profile with lots of different things going on that kind of hit all your different kind of taste receptors. So it might be that your body is quite good at picking up something that it thinks is going to be energy dense and also kind of really rich in kind of amino acids and vitamins and all these different things that your body's going to need. But then also thinking about the kind of bitter taste, and like your body is really kind of tuned into kind of bitter flavors. So it might be that we only taste whether it's sweet or not sweet, but there might be a whole range of bitter and savory flavors that our body's much more kind of tuned into for evolutionary purposes to go, this is a dangerous thing. So if I'm going to eat my 243rd apple pip, I need to stop eating that soon because that's going to make me really ill. we've got a question. We've got a comment about bitter melons and sort of some people liking, some bitter flavours people might really enjoy, and that might be a just a personal preference thing. And again, it's sort of Marmite, kind of you either love it or you hate it. So maybe some people, sort of the flavour profile that people respond to is going to be different. There's a question here: why does unsalted food have less flavour than food that has been salted?
I mean, this is probably down to the salt in its case, but in terms of salt can change our sort of, when I talked about chemical environment and pH, the salinity, which is how much salt is in there, can also change chemical environment. So it could change which reactions occur in the cooking process as well. But I think generally the salt will it will bring out more of the other flavors that are there, and you will notice them more, probably is what is going on there. Having, you don't really prepare food without salt you don't intend to prepare it without salt, unless you're trying to for a low salt diet. So I've probably never really thought about it too much, but that's what I think is probably going on there.
This is the noticing kind of factor. Okay, we've got one little question about sort of the health risks of food, probably moving on from our cyanide segment, and the ideas and thoughts on MSG, so sort of like a super salt and monosodium glutamate. I think that's just a huge flavour enhancer. I mean, I'm not sure I'm a big fan of kind of ladling on more MSG to make something tasty.
Yeah, I think in terms of, it's difficult with these things. So I've got in the sweet section next week, I talk about different sweeteners, and you look at them and you think, well, these are all sold to us as bad things, but you need a lot less for the same flavour. So it's kind of difficult with the balance. I've not really looked into MSG and the exact sort of science going on in there, but I think that you probably need quite a lot less. But I think it also has this weird kind of thing, and people don't like adding additives to their food, but you've kind of got to think, we add salt quite happily. And I think it's sort of, people have this kind of idea of it's a chemical, and yeah, but everything's a chemical, so is salt. But yeah, I think I've not actually looked into it too deeply. I think everything in moderation is sort of the way with food. So if you have lots and lots of Chinese takeaways with lots of MSGs in them, it's not going to be good for you at all. But that's not just going to be down to the MSG, it's going to be down to everything else that's in there.
But I suppose there are all sorts of different ways to enhance our flavours by even just things like herbs and just adding fresh ingredients and cooking things ourselves, or maybe even experimenting. I've got a little allotment plot that I've talked about before, something on the windowsill, just growing a bit of basil or marjoram or something like that can really boost the flavour of the food that you're cooking. I think we're probably close to the end of the session, but next week we're going to explore the sweet end of the flavour profile. And I've just got to whet people's appetite. There's some little multicoloured macarons, and we might mention macarons. These are hideously expensive, and Jess is a bit of an expert in making sweet things, so we're going to explore more of that, and maybe Jess will tell us a bit about her time in Paris, working as a chef, or working in a cookery school in Paris next week. But I'm going to leave that with you hanging. Jess, thanks so much. Thanks everyone for the comments and the questions. I really liked stereoisomer is a great word. I agree. And phenolphthalein is a good word to spell. Not so much fun to have it in your body though. It's not only an indicator, it will make you your bowels very runny. I think so. That's a bit of chemistry that you don't want happening with you. But anyway, on that note, thanks so much, Jess, and I'm looking forward to having Jonas back next week from his last-minute wine tasting and hungry session, and we can talk about making this lemon meringue cake. If anyone ends up making a pizza, so I've made my carrot pizza if anyone wants to kind of share some photographs, or if you want to try making the lemon meringue cake and showing us maybe next week, if you have a chance to make that and take a photo, you could send it to the Oxford_if Twitter or Instagram page. That would be great. It'd be good to see what people are doing. And we hope to see you back next week to talk about the chemistry confections of sweet things.
2 seconds, Dane. Somebody, I've just seen in the Q&A, there's a question about why does pizza dough brown when you cook it? Come back next week. I'll talk about why cake browns when you cook it, and it's the same process. So if you're interested in that, I will talk about that next week. And back to you, Dane.
No, that was good. Yeah, we'll talk about that next time. Someone's mentioned it, but we'll go into that. Maybe if you have time, Google the Maillard reaction, but we'll go into that in much more detail next week. Thanks so much. This will be online soon, and we hope to see you at some more festival events over the next few weeks. Thank you. Bye.